This is episode 278 of the Stem Cell Podcast, Stembryo Engineering, with Alfonso Martinez Arias. Hey, everybody. We are Doctors Daylon, James, and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have truly one of my favorite interviews of all time, frankly. Dr. Alfonso Martinez Arias. He's from the Universitat Pompeu Fabra. He's on the podcast to talk about his research on gastruloids and mammalian development. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, we'd like to remind our listeners about ESC and iPSC News, one of StemCell's free weekly scientific newsletters. ESC and iPSC News summarizes all the latest research, news, jobs, and events in ESC and iPSC research and delivers it right to your inbox every Wednesday. Save time and keep current with ESC and iPSC News. Subscribe for free at www.escellnews.com. Yeah, it was a fantastic interview with Dr. Martinez Arias. It was one of my favorites. I'm with you. He has such a great history in that particular field of embryo modeling and developmental biology. It was cool to just kind of recap that history, and it was like story time. You remember story time with Irv? It was kind of story time with Alfonso this time. I'm a fan of story time. So, anyways, moving on to the roundup. First paper here is going to be from another former guest of the show and someone who needs no introduction. This is Hans Clevers, the one and only organoid maestro, whatever you want to call him, over there in the Netherlands at the Utrecht, but also he's been affiliated with Roche as well. So the first author here is Lulu Wang, and this is a Nature paper that's looking at, surprise, surprise, the human intestine, which is really Hans's tissue type of interest over the last however many years and decades. The title of the paper here is Tuft Cells Act as Regenerative Stem Cells in the Human Intestine. And, I don't know a lot about tuft cells, but it's a population that has been described as this apparently long-lived, quote, post-mitotic cell type. And the question is, and we were talking about this before the show, if they're post-mitotic, how can they be referred to as stem cells, right? And that's kind of what they're getting at here. And they found that they actually have this ability in the intestine where they can transform into stem cells. That's pretty cool. Pretty powerful. Anytime you're describing a new function for an existing cell type, I think that's going to be a pretty high profile paper. And of course, the Clevers Lab and the Clevers Group, team, empire, whatever you want to call them, they were using organoids as their way to identify what these tuft cells are actually doing and how they're serving as this regenerative population. So there's two distinct subsets of tuft cells, apparently, tuft 1 and tuft 2. And what they did was they analyze primary human intestinal resected material and also intestinal organoids, of course, like I mentioned. And I actually ended up identifying 4 distinct human tuft cell states. It's more than just the tuft 1 and tuft 2. And 2 of those overlap with their mouse counterparts, okay? And they did a bunch of deep analysis and actually show that the development of these tuft cells depends on Wnt, not so surprising, and that the numbers of these cells rapidly increase on exposure to IL-4, interleukin-4, and interleukin-13, as folks previously reported in mice. So that's kind of confirming what's already known. And that was through the proliferation of existing tuft cells rather than the increased de novo generation from stem cells. Now, the cool part of this was the second half, I think. So it turns out that proliferative tuft cells, what previously were not expected to be proliferative, they actually do occur both in vivo, in fetal and in adult human intestine. So they're going straight to human here. So they are able to find that these tuft cells indeed do divide. And these single mature proliferating tough cells, just single cells, can form entire organoids, human intestinal organoids that contain all the intestinal epithelial cell types. So this is a big deal. I mean, this is describing a previously unknown function for these thought to be post-mitotic tough cells in a human context. They can regenerate the entire intestinal epithelium, apparently, from single proliferating tough cells. Very powerful. And unlike, true, what they call true stem and progenitor cells, although it seems like these kind of revert back to stem and progenitor cells in a capacity, right? These human tough cells can actually survive, get this, irradiation damage, which has a lot of implications there for cancer. Obviously, chemotherapy and radiation in the context of cancer can wipe out a lot of stem cell populations, but apparently these tough cells are resistant to radiation damage, and still retain the ability to generate all of the other epithelial cell types. So I don't know if you want to call it like a transdifferentiation or whatever, but these are exceptionally powerful cells that even in the context of a brutal environmental stimulus like radiation can still restore the proper cell populations in the intestinal epithelium. And then, they took this even to another level. They engineered their intestinal organoids to actually lack these tough cells. And as you might expect, based on what I just told you, those organoids without the tough cells did not recover from the radiation induced damage. So it's almost like these things are providing the intestine with some sort of radiation shielding or the ability to restore itself after such a brutal insult like radiation. So ultimately, they're describing these tough cells as a damaged, induced reserve intestinal stem cell pool in humans. It's like a backup stem cell population in contrast to those standards like LGR5 positive stem cell populations that Hans has described in the intestine for a long time. It's like a secondary backup stem cell population that has anti-radiation superpowers, it seems like.
Yeah, super cool. I mean, they might want to drop the T off the end there because these tough cells are tough. Ha ha.
I like that.
The many things about this paper. Number one, Cleaver's back at it again. Not surprising there at all. But the surprising thing is the dipole here, I guess you call it. It's like, oh yeah, tough cells, post-mitotic, who cares? They're nothing. Oh no, sorry, they're everything. And a great illustration also of the power of the organoid, from this, maestro, as you call them, organoid maestro, and still, developing these innovative, elegant approaches here in the case of the radiation. I love that element of engineering the organoid with so they don't have the tough cells and not being able to respond from the radiation damage. A really nice and again elegant illustration of that potential and that essential quality. And the last thing it reminds me, I forgot the investigator, so my apologies, but in the ISSCR this last meeting, there were these hillock cells. You remember there were these stories about the lung hillock
Yeah.
cells?
Yeah.
And again, there also I was like, wow, look at these robust cells that can seem like you could dump acid on them, and yet they come back at you. And it's not perhaps, I mean, it is surprising, but there's a nice agreeable symmetry there to the idea that there's a correlate in the intestine, and perhaps if you extend it in other organs, there may be these tough cells, tough TUF cells or TOUGH cells, whatever you want to call it, but cells that are really robust in there specifically to recover from insult.
Yeah, really powerful. I like how you describe them as tough cells because they are tough cells. That's what they're doing here. They're restoring the intestinal epithelium where everything else gets wiped out by something as intense and brutal as radiation, right? It's also a reflection on something we've talked about a little bit more here on the show, this idea of cell fate plasticity. And we're going to have an upcoming conversation with someone on the show specifically about this topic. But this idea of transdifferentiation in vivo may be more realistic and more viable than you might expect. Because that's kind of what these things are doing. These are in the presence of certain immunological signals. These tough cells are transdifferentiating to other epithelial cell types purely in vivo. It's a concept that maybe wasn't as hot and interesting 10 years ago, but now it seems like there's major papers coming out all the time that's describing this process in humans, right? This is a human system, these human organoids. Um, Things are turning back and forth into different cell types all the time, it seems like.
Yeah, again, the dipole, it's probably not the right word, but you get the idea. We used to think about terminal differentiation, like how far we've come, right, in terms of plasticity. And you're alluding to a conversation that we're going to have in a couple episodes with Sam Morris, which we're really looking forward to. We got to chat about this idea of transdifferentiation and cell plasticity. Really looking forward to that. Moving on from the maestro of organoids, the originator, to like maybe the apotheosis of organoids here, and perhaps not. I mean, the story keeps on winding out. So I'm sure we'll see more complex organoids to come. But here is a story about this pretty complicated organoid, which is being used to glean insight to hematopoiesis, right? Definitive hematopoiesis. Just brief review. There's a primitive hematopoiesis that really only accounts for the limited repertoire of erythromyeloid cells, and just disappears pretty much by a month in of human development. And then there's that definitive wave, which accounts for the rest of hematopoiesis. Specifically, accounts for hematopoietic stem cells. It has this intraembryonic origin in the hemogenic endothelium in the lumen of the dorsal aorta, right? Which is at that point within this aorta gonad mesonephros region, between 4 and 6 weeks of gestation. And the niche there that supports this definitive early hematopoiesis is elusive. We don't really have a way to see into that, obviously, in human embryos. And it's really a key goal to understand that, because I think a lot of consensus is that part of the reason why definitive true hematopoietic stem cells from pluripotent cells has been such a long sought and challenging goal is that the niche is so transient, and establishing a niche that can support that cell once you induce it or differentiate it is really the challenge. So this is an effort to try and understand that niche, but it's complex, right? There's a lot of components there. And, there's one endpoint here, which is generating definitive hematopoietic stem progenitor cells, which you might argue we've already got there, right? We've differentiated hematopoietic stem cells to a lot of hematopoietic, or human pluripotent stem cells, I should say. We differentiated them to a lot of hematopoietic derivatives. I was delighted to see that this story, which is from the Zweigert lab, with also lead author here, Lika Draklis, in Hanover Med School in Germany. I was delighted to see that they referenced my first senior author paper, which is over 10 years ago now, so I'm aging myself a bit there. But the point being is that we've gone some ways in differentiating hematopoietic cells. Definitive remains to be seen. I mean, there was that paper we just talked about briefly from Stanley and Alfanti labs, where they made a definitive engraftable cell. But remains to be seen whether that's validated, although I believe in it. But the key there is that even though the endpoint was really impressive, how they came about was a bit murky. It was a diff protocol. They took the output CD34 cells. The key is how do we generate these? What's the niche within which they form, right? So this was an extension of a previous story where Lika Draklis was lead author. We covered it on the podcast actually about these heart-forming organoids from the Zweigert lab. Lead author Dr. Draklis there, and here in this extension, they made these blood-generating heart-forming organoids, right? By extending the diff and making it more, modulating the differentiation conditions. And here, talk about superorganoid, they got co-development of a lot of heart components, cardiomyocytes, also septum transversum, or these proepicardial cells. Also, arterial and venous endothelial cells were in there, hemogenic endothelium in there, hematopoietic progenitor cells, and their derivatives, mature hematopoietic derivatives. So, and those derivatives were reflective of multiple waves of both primitive and definitive or lymphoid potential hematopoietic cells. And also anatomically, in terms of how they looked in these organoids, they were, reminiscent of that anatomical formation, and they demonstrated the multipotency that you would see from these hematoendothelial cells from pluripotent stem cells. Now, no engraftment per se, but I think this is a different kind of study. We're not focused just on the endpoint, which is the holy grail and is a big deal. It's more about generating these proxies for human development that are all the rage now. And I think the most exciting thing as a root, developmental biologist, that's what's so cool about being a scientist in this era of stem cell research, is finally having our hands on these organ homologes in human.
Yeah, this is really cool. And, side note, it's always fun to see the work that you did many, many moons ago be referenced in such a seminal paper. It's the best feeling. It warms your heart every time. This is a great paper, just an extension of some of the amazing work that the Zweigert Lab in Leukodrochlis has been doing over the last few years. First author here is actually Mariana Dardano. I actually, we're dabbling in cardiac organoids in my own lab. And we refer to the Zweigert Lab's expertise and their previous papers all the time. I actually have to honestly think if there's one lab who's leading the charge when it comes to cardiac organoids and making the most advanced cardiac organoids, it's the Zweigert Lab. I think they are the best in the world. So shout out to the Zweigert Lab for their amazing continued work here. I mean, they're just taking these things to the next level. I mean, initially they just described those really advanced heart organoids, but now they're incorporating this, hematopoietic generation, the system in there as well. I think, what you're alluding to is really important. It's the presence of the niche, and we discussed this on the show before, the importance of the niche in helping to create these subpopulations of cells. You have not just cardiomyocytes, but also the hematopoietic progenitors as well. And I like how you brought up the Elefanti paper as well, the Nature Biotech one that we covered briefly a couple weeks ago. I mean, that is a diff protocol that's trying to make, quote, bona fide HSCs for ultimately for transplantation purposes. And there were limitations in that paper because they didn't do a great job with the secondary transplantation in the mouse, like we talked about. But maybe this is the way to do it. I mean, that is first and foremost a diff paper. This is not a diff paper. This is more of a developmental biology paper. But perhaps you can harness. Maybe this is just another way to make bona fide HSCs. You make them in the appropriate niche using this developmental system, and then somehow you extract them or harness them and scale them up ultimately for therapeutic applications. I'm sure it's harder to do in that context because you don't get a ton of cells when you do it this way. But maybe they're more bona fide when you incorporate the appropriate niche in which they're supposed to be made, right?
Absolutely. And yeah, I was just thinking the same thing. Maybe scaling would be tough when you're going on that organoid level. But just exactly what you're saying. You look at two different things. You have the pragmatic approach, just getting the cells at scale, but also understanding the niche so you can support them and maybe like increase the scale at which you generate them in those, less organoid or organ proxy conditions. But the engraftment stuff remains to be seen. That's the real question with these. The hematopoietic progenitors that come out of these blood generating heart forming organoids do a lot of stuff, busy. Will they engraft and will they long term? And another thing I love, and I think this is an editorial, and of course you don't know what went on behind the scenes, right? Maybe this paper was submitted 6 years ago to Nature and they waterfalled it to Nature Cell Bio, where this is. But if you look at the publication data, this was received January 2024. So I like this, and this is a shout out to the editors at Nature Cell Bio, who we love. um, that the editorial process here was I think I wouldn't say rushed, hasty. I thought it was appropriate. And whereas they could have asked maybe for that engraftment stuff in the primary and secondary, which would have taken months, if not years, I think it's great that this paper is out and people can build on it. And it's definitely nucleating a lot of alternative pathways. Maybe people wouldn't have thought of hard argonites for their hematopoiesis, but here we are.
Yeah, absolutely. And of course, shout out to Stelios Lefkopoulos, our friend there over at Nature Cell Bio, who we had in the show, one of the editors there. They're doing really great stuff in that journal. I love looking into what they're doing and seeing their cool organoid systems that are being published there. It's not just Stem Cell, it's Nature Cell Bio. It's all these great journals that have a broad range of exciting work coming out in the realm of organoids. Moving on to someone else who doesn't really need an introduction. This is Anne Brunet at Stanford, who I've gotten to know over the last few years when I was there. This is a Nature paper coming from her lab, and her lab is really focused on all things aging, and that's been her bread and butter for many, many years now. And it seems like this is one of the hottest topics recently in stem cell biology is using different approaches to address this thing that we're all going through, which is of course aging. And we have talked about papers from Altos Labs and Juan Carlos Belmonte using the OKSM to reverse potential aging phenotypes, which is still a bit of a stretch. And I think they're rightfully investigating that in the context of basic biology right now before ultimately putting it in people. And that's what they're doing here too. In this Nature paper, they're looking at basic mechanisms of aging, and specifically aging in neural stem cells using a really powerful CRISPR screen. So that's pretty much the title of the paper. CRISPR-Cas9 screens reveal regulators of aging in neural stem cells. And the first author here is Tyson Roots, like I said, coming from Anne Brunet's lab, but also on this paper are Michael Bassick and a few other folks from that I'm familiar with at Stanford. So aging impairs the ability of these neural stem cells. And this is, as we've talked on the show with folks in this field, this is a paradigm and an area of study that has rapidly changed in the last couple of decades. A while back, a couple of decades ago, when I was in high school or whatever, elementary school, grade school, this concept of neuron, post-birth neuron generation was a new one, right? Folks were kind of not into it. They were not convinced by it that new neurons can be formed in the adult brain after birth. And now I think it's a pretty well established thing that you have these neural stem cells, which ultimately do make new neurons in the adult human brain in the right context, in the adult mammalian brain. So, but the problem there is that these neural stem cells age, they degrade, they lose their potency and transition from proliferation to quiescence over the course of aging and during the adult mammalian brain. So this functional decline, like I said, results in the decreased production of new neurons and defective regeneration following injury during aging. And there have been a lot of genetic interventions that have been found to ameliorate or improve old brain function, but you really got to dive deep into the NSCs because that's the key here, right? In terms of generating new neurons, you have to systematically, functionally test different genes in old NSCs and more generally in old cells. That's kind of what they did here. They did a pretty brute force CRISPR screen, high throughput in vivo and in vitro CRISPR screen in mouse in particular, using CRISPR-Cas9, which is Cas9 is a bit of an old school technology at this point, but it's so well established that I think it is still the go-to for CRISPR screens. Using the screen, they did a bunch of gene knockouts and figured out which genes, when knocked out, were able to boost NSC neural stem cell activation in older mice. And they found that in these primary cultures of young and old NSCs, they uncovered more than 300 different gene knockouts that could restore the function and restore the activation of old NSCs to actually create new neurons. The top gene knockouts were actually involved in cilium organization and, importantly, in glucose metabolism and glucose import. They established this scalable screen also in vivo, like I mentioned, took their in vitro and brought it to the mouse in vivo screen, which identified 24 knockouts that were boosting NSE activation, the production of new neurons in old mouse brains. And in particular, they focused on the knockout of SLC2A4, which is a gene that encodes for the GLUT4 glucose transporter. I mean, that's a pretty big deal. That was the top gene, one of the top genes that improve the function of old NSCs and their ability to generate new neurons. So there's glucose uptake is increasing in neural stem cells during aging, and transient glucose starvation apparently restores the ability of these old NSCs to activate. And that's important because you can see that's an intervention that's not genetic. All you have to do is potentially do some sort of glucose starvation, and you have an increase in proliferation of NSCs and the activation of their function. So, an increase in glucose uptake may contribute to the decline of NSC activation with age. That can get a lot of people's heads turning, right? In terms of the connection of there is like what you eat could be very important to the aging of your neural stem cells. I think it's a stretch, but I think that is what they're alluding to here. And that's, I think, just one of the results, one of the findings that they got from this awesome CRISPR screen. So, for one, it was establishing the platform, a scalable platform to actually identify genetic interventions that can boost the function of these old NSCs and bring this platform in vivo. But two, it's the implications, right? Important implications for maybe countering regenerative decline during aging, maybe just by taking up less or eating less glucose, maybe not eating as many candy bars will do the trick.
Caloric restriction, here we come. I don't know. I don't have the strength of mind, our body, to be honest. My neural stem cells have already declined. Woe is me. But I think you're right there. The reason why this was a Nature paper is that element of kind of independently validating the mechanism, because the power of this is the scale, right? The tool, you can do multiple genes, and that's great. I can't underscore that enough. I think, because I'm going through some data recently from a RNA-seq. And you know how you've been there, you review these data sets, you scan that Excel sheet, and you got to look at, thousands, tens of thousands of lines. So what jumps out at you is the things you already know, right? You find what you're looking for. Yeah, you certainly don't find SLC2A4. That wouldn't perk me up at all. It doesn't sound familiar. So these types of screens, I think, are really important for zeroing in on things that otherwise would go under the radar. And here the approach, though, kind of calls into question, like, oh wait, that's counterintuitive. Knockout to restore function, but it's not unprecedented. There's plenty of cases where knocking something out is actually the therapy, even though it's a hack. So like that makes sense, but then they close the loop there by going in on this mechanism with the glucose uptake. And I agree with you, it is a bit of a stretch. And the metric of neural stem cell activation, while that is coincident with neural decline, reduced activation and proliferation, you don't know exactly in terms of like function, or I don't know, synapse, neural network, who knows? Proliferation isn't the only metric. So there is some ways to go here, but like intuitively, while at the start of it thinking knockout can't be helpful, once you get the whole story, like, oh yeah, and then you're like, oh wait, caloric restriction, then you're like, here we go. So I can see why it's a Nature paper, and I really appreciate how they told a beautiful story here, starting from a large scale, open-ended, kind of unbiased platform.
Yeah, this is old school genetic screening brought to the modern CRISPR age. And knockouts, as you know, are the lowest of low hanging fruit when it comes to genetic screening. It's easy to do a knockout, right? You just throw up some Cas9 into the gene, throw some guide RNAs, and just take that shotgun approach to the gene and just destroy it. Really easy to do as opposed to the opposite, which is the overexpression. And I would love for them to do that screen down the road because CRISPR-A is also a thing, of course, right? And maybe that's something they're thinking about. But I think this is the most straightforward approach, and maybe that's just the next phase in this uh, search for things that can regulate NSC function.
Agree. Agree also to, yeah, the easiest thing to do is to break a thing. Sometimes that can be therapeutic. Sometimes, rage. Not really. But I'm segueing here into the tough thing, as you're talking about, is to build a thing, right? And we're going to talk with Alfonso about his seminal work in building these gastruloids. And as an entree to that discussion, I have a story again back in Nature Cell Biology. This story from Princeton, led by Harold McNamara and Jared Tocher. And this is a story about, gastruloids and symmetry breaking specifically, which is something that happens in the course of getting a spatially organized structure. You need to undergo symmetry breaking, or else you're like, like a sand dollar or something. Even they break symmetry, so you got to do it to be a thing. And in the embryo, symmetries are often broken by these extrinsic cues, right? Which provide like an overlay of positional information, this coordinate system in the form of like morphogen gradients, for example, right? And using that coordinate system, these relatively simple aggregates of cells can build embryo-like structures. And this can happen even in vitro, without that full complement of the morphogens. And here, I mean, apropos to the title of this episode, I hadn't heard this term before, but here the authors refer to these as these Stembryo systems, which is these embryo models that we commonly work with, or gastroid models. Arun and I were laughing about this. Stembryo. How do you guys feel? You should leave a comment. How do you guys feel about the Stembryo as a moniker? I feel like Stembro's next. All these millionaires coming out of the stem cell industry. Stembryo is next. But here we're talking about Stembryos, and these things offer a window into the mechanistic basis of stem cell self-organization. And one of the most important critical events there, as I already mentioned, is this overlay of the coordinate system. So you can define body axes, right? And that's in the embryo, governed by, or during gastrulation, by morphogens, right? And also the inhibitors. And that's how you get these specific signaling domains, right? Cut to mouse, in this case, gastruloids, right? Mouse gastruloids, they break symmetry and form this anterior-posterior axis, but not in the context of, this is in vitro, seminal work. They do this not like in the context of all these morphogen gradients, although that is coming to the fore. Now a more ordered, like morphogen gradient driven differentiation is happening. It's really exciting. But like these gastruloids, they do it spontaneously. You just put them in shear 99021 for the 24 hours between 48 and 72 hours after they're aggregated. And they undergo this profound morphological, but also signaling changes. Including like the formation of this brachyury positive posterior pole, where Wnt signaling is activated. But it's not clear how does, in the absence of like all these overlays, which are really well studied in the context of like mouse, other mammal development, but not in humans so much until now. How does this happen when you just have like a uniform cue, just shear, just Wnt activate? How you lose all those other stuff, all those other morphogens? Right. So here, in this story again from McNamara and Tocher Labs, they engineered these really cool synthetic signal recording circuits that were based largely on this Wnt responsive element promoter driving a lot of reporters, in combination with like CREAs and the stop blocks, lineage tracing, where it turns from tomato expression to, or both tomato and GFP, cuts out the tomato, just GFP. So all these lineage tracing they use to label the cells according to their signaling state within these defined windows, focusing here namely on Wnt. And they show that that there's early domains of Wnt activity that predict the position of the cells along this AP axis, and that's before the actual Wnt signaling pattern has even polarized. So the activity before polarization predicts the position, and that polarization it happens by way of this rearrangement, sorting of Wnt high and Wnt low cells. And the kicker here is that the nature of that Wnt high or Wnt low is dictated by this pre-existing response to nodal and BMP at an earlier date. So like there's all this history of these cells that the authors here were able to elucidate with their technical approach, which showed that this programming, the sequential programming of all these signals, enables this self-organization without extrinsic overlay. And this is really cool. I mean, from a basic mechanistic standpoint, but also like in terms of generalizing this approach, going beyond like gastruloids, I think it raises the prospect, because let's be honest, like none of these embryo models, they all lack extra embryonic components and integration with the circulation, etc. None of these are the real thing. So everything's a hack. And the question is like, what are we able to create? And I think this study and the approach here, it gives an idea of how can we create just organ enlages across the board, to try and actually generate, I don't want to say organs, but at least model organogenesis in a way that is kind of de novo from the origins of that organ. Don't have to start from the gastruloid. So I thought this was a really cool, technically, also like in terms of the conceptual advance and innovation.
Yeah, really cool stuff here coming from the Department of Molecular Biology at Princeton. We don't honestly cover a lot of papers coming from Princeton, even though it is such a well-regarded academic institute. I think, they're well known for their ability to dissect basic mechanisms of molecular biology. That's been their power over the last however many years. And then incorporate bioengineering techniques into dissecting those mechanisms. You don't hear a lot of like translational stories coming from Princeton, for one, because I don't think they have a med school. I think Rutgers or something is they're affiliated with med school, but they do basic science and basic biology really, really, really well. And this is incorporating a really neat synthetic bio approach to figuring out the basic mechanisms of how development works in these gastruloid systems. I wouldn't be surprised if other folks, do similar things. I know there's a great team at UCSF who's doing similar synthetic bio approaches to open up that black box of embryo development. So I think this is incorporating a bunch of technologies that we're familiar with here on the show, and ultimately that's the goal, is to open up that black box, right?
Yes, you said it. The Princeton is great at this stuff. The synthetic bio, the systems bio. I remember Ihor used to work there, and that's where he kind of cut his teeth. Ihor Lemishka. I remember Arnold Levine, who was the president of Rockefeller University when I was there. He came from Princeton, or he went to Princeton. They had the Institute for Advanced Study. Yeah, they're about like basic and deep thinking, and it shows in this paper. I mean, you're looking at all their systems and like the tracing and the machinations they take in order to get the insight they do, give you a headache. But they have the capacity for it. They got those neural stem cells still firing, unlike me. And this really was a great illustration of that. And also, as you're alluding to there, a kind of a sign of the times here, where this synthetic biology is really coming to the fore. I mean, the AlphaFold and protein engineering just won the Nobel. And I think it's this type of study, although not like directly related to that, that this notion of synthetic biology as a means of mechanistic insight and study platforming, but also to create cells that can be applied, or proteins or whatever, to create biology that can be applied toward whatever use. I think that's the era we're entering right now. Very exciting, a little bit scary. But all our stories today, nature, and nature, I think really covered the four. And we're going to extend that conversation with Dr. Arias in just a minute. Before we get there, I got a quick message from STEMCELL Technologies. Do you want to implement the new ISSCR standards for human stem cell use in research? Get a free checklist wall chart that helps you follow the recommended reporting practices and quickly reference the relevant sections of the standards. To learn more, visit stemcell.com/ISSCR-standards-wall chart. All right, everybody, with us for this episode, we have Dr. Alfonso Martinez Arias. He's a research professor in the Department of Systems Bioengineering at the Universitat Pompeu Fabra, and he's been doing developmental biology, a lifelong developmental biologist. I don't want to age him, but he's been doing it longer than he hasn't. I'll say that much. But in recent history, over the last 15 years or so, the Arias lab has focused on using embryonic stem cells to explore cell fate decisions during mammalian development. And for the last several years, they've been using gastruloids to explore a number of novel research avenues about how cells form tissues and organs that cannot be explored using more traditional models of embryogenesis. Dr. Arias, thanks again for joining us on the show today.
Pleasure to be here.
The pleasure really is ours. You've been working, as I said, in developmental embryogenesis for, I'll just say it, more than 4 decades. I aged you a little bit there, sorry. And you've worked across all the model systems, not really all of them, but a lot. And you most recently, I love this, the symmetry here. You kind of close the loop in a way by reconstituting aspects of human embryogenesis, because that's the goal for all developmental biologists, right, is to approximate the human product. You've kind of reconstituted a lot of aspects of that using pluripotent stem cells. So why don't we start with you telling us in your own words about what your lab is focused on these days?
Well, as you mentioned, it's about, as always, time passes quickly, and the pandemic created a funny gap in our lives. And it surprises me that it's over 15 years that we started working with embryonic stem cells. It was at the beginning of the 2000s. I had been working with Drosophila for many years, but I suspected that Drosophila is an organism which seems to be very determined by the genetics. There is very little of self-organization. It was an organism that was selected because of very fast developmental mode and because they were looking for mutations. However, hidden behind that genetic determinism, I sense, particularly because of an interesting Wnt signaling that I've always had, that there might be other things lurking in the background. I was also always fascinated by the experiments of Hans Risch, where he separates blastomeres, and he manages to obtain whole embryos from separated blastomeres, or that you mix blastomeres and the different embryos and they manage to get an embryo. So I was looking for a system that would allow me to explore those features of development, because I've always had the impression that the most important feature of development is the self-organization, is the ability that cells have to talk to each other and to characterize. Then, as you know, and it's the essence of this program, stem cells came in the late 90s as a very interesting system to do regenerative medicine, but also we and a few others in the UK felt to do developmental biology in a new manner. And that's how we came to start using embryonic stem cells to do developmental biology. To me, the most fascinating aspect of the stem cells were that you could differentiate them in vitro into any cell type of the organism, but they would never make the organism. I mean, to me, that was a bit of a puzzle, more so because if you put them back in a mouse, and this was, of course, the embryonic stem cells of the mouse, they would contribute to the mouse in a very interesting manner. Mm-hmm. So I felt that either there was something mystical about the embryo, that we're missing something. So that's really how we came to start working with embryonic stem cells, to learn about the early stages, the pluripotency, the exit of pluripotency. I have to say that we were incredibly lucky that at that time Austin Smith had moved to Cambridge from Edinburgh, and he was extremely helpful in drilling us in how to grow embryonic stem cells and how to do good culture work. Then it's a course of failures until we surprisingly ran into this system of the Gastruloid, which are proving very successful, and I'm sure that we'll talk more about it. But what I'm grateful for the 2 or 3 years that we spent working on pluripotency under the very careful eye of Austin and his people, it is becoming clear, and we hear that in this program very often, that the way you culture, the initial culture conditions are critical to what the cells are going to do.
Yeah, that time in the 90s and 2000s must have been such an exciting time to be in the lab, and just figuring out how these things actually grow. And I think some stem cell biologists, maybe of my generation, take this for granted, right? Back then, just getting embryonic stem cells to live and grow and multiply and survive was such an accomplishment. And now we're just doing whatever we can with them, right?
I think there it's interesting the difference between the mouse cells and the human cells. Mouse cells are a very, very well catered system, very well behaved. And I think we owe that to Austin Smith, who I think worked very, very hard in creating conditions that we can control. When you move to the human ear cells, my main argument is that they're at the mercy of the people that sell you the media. And I think that is I think clearly by your reaction, you know what I mean, and they change the batch and your experiment changes. And that drives me mad. Like, I think many people, I think this is a criticism I have of the field. They don't put enough emphasis in telling us what they do and when it doesn't work. But working with mouse, it's a delight. I like to call them the ideal gas of developmental biology, the embryonic stem cells. But that's because we have those conditions very, very worked out. And I know that Austin and his group have been working also in developing similar media for the human, and it would be good.
Yeah, absolutely. I like that. The ideal gas. I never heard that analogy before. That's a good one. Since you alluded to it, the recent work that you've done on gastruloids has been so exciting. Fast forward a couple of decades and now here we are, right? Really studying human embryo development with these next generation models. You had this landmark study in Nature a little while back describing these human gastruloids as this in vitro model of anterior posterior organization during human development. And I mean, listeners to our show are very familiar with these gastruloids. We talked about them a lot, the work of yours and as well as other folks around the world as well. But in this particular study, you first described them as these 3D multicellular aggregates that can differentiate to form derivatives of the germ layers, and they're organized spatiotemporally. So that's super important without adding additional extra embryonic tissues. That's also really important. So I'm always really curious about the work that goes on behind the publication to actually make something like this happen. So tell us more about these, human gastruloids. How long was this in the works, and what did it take to make them a reality?
So I think we need to start in the mouse always. I think people sometimes underestimate the importance of the mouse. And between 2012 2014, we developed the model in the mouse, which was a surprise. We were trying to make embryos, but we were not. It's not easy, as you know, from the stem cells with the different extraembryonic tissues. But then we decided, for work that we were doing with brachyury in adherent culture, to try the cells on their own, and we were surprised to create that these structures appeared in the plate. They were very surprising, and the key thing is the initial number of cells, and this is something that still surprises me. This is the thing that determines whether the cells are going to create a gastruloid or an embryoid body. We were very surprised. It's a very narrow number of cells. If you go below 200, it works stochastically. If you go above 300-400, you get an embryoid body, just like getting an embryoid body. But within that range, the cells seem to be able to self-organize themselves and organize their genetic programs. So I think the basis we spent, when I saw this, I was a bit concerned that it might be just some epiphenomena of the aggregates of ES cells. But when, in a very fruitful collaboration with Matthias Lutolf and Denis Duboul, we started to see that there was an inner structure that was very surprising. After many years, and I may want to come back to what we are learning from the mouse, because they are the more advanced, the more that people are using. One of the most interesting things of the system was that either it works or doesn't work. I mean, I have a lot of criticisms to many of the embryo models that are done for post-implantation development, because they happen at such low frequency that it's almost a fluke that they happen. But in this system, it was very interesting. It's not like one day it's going to work 20% and the other is going to work. If it works, either it works or it doesn't work. And I think there is a biophysicist at Pasteur, Thomas Gregor, who has been using them, and he gets 99%. I mean, this is so that's what makes them very useful. If you control the initial conditions, it works. So once we started to see what this was and what it meant, and there is a lot of interesting things to talk about, then we decided that we should go into human. Working with human was a bit more difficult because, of course, the human ES cells are not the same as the mouse ES cells, so we had to adapt protocols. And here, very important, the work of Naomi Morris, who was a research fellow in the lab and now is a PI in the CREA. And she managed to simplify a protocol that I and a technician had started with that was very complicated and very haphazard. I think that's also for people to see the gastruloid protocol, both mouse and human, is very simple in the beginning. Behind that, as I think you've said before, there is a lot of trickery and a lot of tinkering. So I think that it's a very simple protocol. We are very surprised. You basically steer the cells out of pluripotency. You put them in a stage where they are about to start gastrulation. And now we know that very, very well from both the human and the mouse. And then all they need is a pulse of wind, which is, to think about it, it's exactly what the embryo does. And you give them that pulse of wind, and then they go, because you've set them up for some reason. And the crucial situation is that early stage and the number of cells. And this is something you were asking me, what are we looking in the lab now? That's one of the things that we are mesmerized by. What is it about that number of cells, which happens to approximate the number of cells with which the embryo starts gastrulation? How are the cells reading that? In fact, there is beautiful experiments by Patrick Tam many years ago, where he tries to change the number of cells at the outset of gastrulation, and he observes that the embryos wait for the right number of cells in order to gastrulate, which is a very fascinating paper that he does. And Patrick Tam is, of course, the great embryologist, the mammalian embryologist. So that's the way we came to the human gastruloids. Because they don't look like embryos, a lot of the work has been to try to understand what they are. And perhaps the more fascinating thing is that they have a very good spatial organization of the genetic programs within the three axes, which to me is the main issue of gastrulation, but they don't have a lot of morphogenesis. These things are growing in suspension. But if we put them in matrigel at the right time, all of a sudden morphogenesis appears. And there have been many somatogenesis-derived organoids from our gastruloid system, and they all are based on the same thing. It is remarkable if you think about it. I mean, what have we learned? Many things from the gastruloids, but one of them is that the genetic program and the morphogenesis are devoid from each other. And this is very important. You can have very nicely spatially localize the whole program of somatogenesis, but no somites. So you have a region where all the genes that are expressed in the somites are being expressed, but there is no somites. You put matrigel, and the somites, bang, they appear. That's a very remarkable thing. And it's very, very important to understand this, I think, because this is telling us that there are two processes that you can separate and that we might have to tackle together in order to get good organs from culture, and because at the moment the protocols they don't seem to appreciate this separation between the genetic and the morphogenetic programs, and that might be important. You know, we are doing protocols that we're not thinking. Sometimes I feel that this field is like alchemy. People are we want the product without wondering how do we get there. To me, the gastruloids, because of this very high level of reproducibility and the many manipulations that we can do on a simple protocol, are allowing us to explore in great detail what are the mechanisms that early in development define cell types and the primordia of organs and tissues.
Wow, what an amazing synthesis. You're so good. And I like how you ended there with, we're all so obsessed with the product. And I think it's what what I'm enjoying about this conversation is the perspective you have from someone who, started in a non-human model system and appreciates all the amazing insights that had to be, all the advances like you described there, which seem kind of intuitive in retrospect. Cell number, what a key thing. But like, yeah, of course, right? Cell number. And like, yeah, in suspension versus with matrix, like, yeah, okay, that makes sense. And nowadays, you mentioned your collaboration with Matthias Luta. Nowadays, we're talking about like bioprinting or like laying down foundations and establishing morphogen gradients. And it's like, yeah, right. And you said it because that's what the embryo does, right? But I've been doing this for quarter century, not as long as you. But since day one, literally the first day in my first rotation as a graduate student, where I ended up doing my thesis, that was like the conversation. The end game here is we're going to establish a tractable experimental model of human embryogenesis. And then I went and squeezed frogs for 2 years getting eggs, and that was as close as we got before we, were exposed to human embryonic stem cells. But it was early days, just figuring out how to keep them alive, establish them, much less iPS cells, patient specificity, all the stuff where we are. We're really here now on the cusp of this long sought goal of establishing a tractable experimental model of human embryogenesis. What do you think is one of the most exciting advances, like in a more material? I know you're about mechanism and like those key modulators, cell number, but like focusing on the product, as you alluded to there. What do you think is one of the most exciting advances going to follow from this field of study? And this is a little bit, asking for a friend, really, no, asking for myself. I know you work in various capacities and applications related to hemogenesis. So I got to ask, do you think we'll be close to this, bona fide AGM-derived hematopoietic stem cell?
I think from the point of view of applications, that's what you're asking. We've been working on 2, but I can see 3. I think that I think there are organs in the embryo that require input from different germ layers from the beginning. I think this is a very different approach from the organoid. The organoid culture immediately steers the cells into a particular direction very quickly from the beginning. But there are organs, the heart is of course the most obvious one, which brings together pieces from many different parts of the embryo. That's going to be difficult to get an organoid, and as has been discussed in this program many times, I mean, you can get the parts, but not the whole. I think what is interesting of the gastruloids is that they give you for free, in a way, all the primordia at the same time in the beginning. Okay, if you can steer that, I think the gastruloid is very good. At the end of the 3rd or 4th day, we have all the primordia there. Now we have to steer them into different directions. And I think with Matthias, we did a rudimentary heart, which they published. And the blood, of course, is the holy grail. We just have a preprint with a number of colleagues, Cristina Pina and Ana Vigas. We were very surprised. We've been working on this for several years. And we are able to generate the niche. I mean, this is something that people can see it. It's in bioRxiv, and we are now in the process of trying to publish it. And by the criteria, Ana Vigas is a well-established researcher in the field of hematopoiesis from the perspective of the embryo. She's and I think we can compare what we think are the hematopoietic stem cells that we can generate in the gastruloid with those of the embryo, and they look very, very similar. So we are very confident. But when we put them in the mouse, and this is without any transgene, this is just playing together with the cells in the gastruloid. But when we put them in the mouse, they vanish. Now, I've been given lots of lessons about hematopoiesis over the last 2 or 3 years. I would say that if I was to be born again and do everything, I might probably, if I had known about hematopoiesis earlier, I would have worked on hematopoiesis. I think it's a fascinating field. And when somebody tells me that about 70% of our cells are in the blood, that makes it even more fascinating. But I think this is a very good example where we're going to be able to use the gastruloid to improve things, because I think these cells look like the ones in the embryo at the beginning, but then we can ask the question, why do we lose them? Is it that there is a heterogeneity there, that the cells have to travel to the liver? Is it that they travel in a very heterogeneous manner and we don't have the factors that are finding? Because if you look at the pictures in the paper, we are getting things that look very much like the AGM and the very, but why are we losing them? I think sometimes also in this field of the organoids, the gastruloids, whatever you want, the regenerative medicine, people don't emphasize enough what doesn't happen. We're all pushed to report the successes. And yet, I think you learn from what it doesn't happen. And I'm very, very interested in this particular observation, that we are getting cells that, by the current gold standard of a cell type, which I don't agree with, by the way, which is the single cell RNA-seq, they look like I mean, I would say that if it looks like a duck and doesn't work like a duck, it's not a duck. I mean, this is something that maybe we can say. Most of the time we get with these cell types is so why is it that they are lost when we would have expected? And this is where we are trying to modify the conditions to try to see what is this niche losing. I mean, this is work that my colleagues are doing. I wouldn't pretend to be someone that understands hematopoiesis, but I think that this is where these organoids, let's call them like that, which are multi-primordia, they are going to be very, very useful. I think the work of Jim Wells that you have portrayed here many times is a very good example of how, if you assemble different pieces, you're going to get a better end product. The gastruloids are giving all of this for free, and it's going to allow us to study what is that and to improve the product. So I see heart. The third thing, I said there were 3. From the very, very early, we had a pilot study with Roche on the possibility of using particularly the human gastruloids for teratogenicity and toxicology. When you think about it, there is no good system that uses approximations or proxies for human embryos in the market, okay? And everybody knows the example of thalidomide, where the big failure was that it didn't have anything to do with animals, but it had a lot to do with humans. In fact, in this work that was published 2 years ago now, we compared the mouse gastruloids with the human gastruloids and showed just some proof of principle that they could be used for this, and there is people out there that are thinking of using. So teratology, these complex organs that have a multi-origin, the heart and the blood, I think those are things that, but I see my job in all this as the basic engineer. I'm interested in giving them very good starting point. Then these people tell me what they want, and I interact with them and guide them into that direction. But we need to understand that early stages, because so many important decisions are made there, and I don't think that it's that we have thought enough about that.
Yeah, I think one thing you're alluding to here is the importance of models that are developmentally faithful, and that's part of the reason why these gastruloids have such incredible potential, because you can ultimately perhaps make derived tissues that are developmentally faithful, whether it's cardiac tissues, whether it's hematopoietic stem cells. Now, with that being said, there's still a really important aspect of standardization. And I think one thing that's really exciting about your gastruloids is that they're so reproducible. Like you're alluding to, 90% plus fidelity in terms of being able to produce them. This is not always the case for other embryo models. And you actually wrote a really nice commentary in Nature Cell Biology focused on this idea of standardization of stem cell based embryo models. I think it's very relevant and very timely. It was co-authored by a bunch of folks in the embryo modeling field, including a number of people who have been on the show, like Nicolas Rivron, Olivier Porquier, Jacob Hanna. I mean, this is something we've talked about a lot on the show. Standardization is so important in stem cell biology. And of course, the ICCR publishes and promotes their own standards and guidelines for stem cell research. And, this commentary was important because it proposed some criteria with which you might be able to raise the standards of these new stem cell derived embryo models of human embryogenesis. So tell us more about this effort, this important recent effort that was published, and what are you hoping to accomplish here?
You want to know the story? I mean, this started a while ago, and partly is by the restlessness of some of us in seeing some of the embryo models, human embryo models that are being produced, that they seem to me to be more the product of chance, many of them, and really uninterpretable. Some of them are very uninterpretable and associated with very big headlines about what is that they have produced, which can run into big difficulties. I'm very concerned sometimes about the effects that those headlines can have on research and have had on research. So some of us felt that it would be very good, particularly I work very closely with Nicolas Rivron in these things. And in fact, we have penned more than one commentary, but this was very important. We felt that we needed to draw some standards. This is a very new field. This is something that people have to appreciate. That the same with regulation. That's another thing that we are very, very involved in. But I thought standards are very, very important. People also talk a lot about blastoids and gastruloids. I think are the most consistent and more workable systems that are at the moment. But in the middle, there is a lot that it was not defined. So I felt that we needed to bring together stem cell biologists, and if you look at the author list, you will find some embryologists, Katja Jantionakis, Patrick Tam, Janet Ross, and I felt that it's very important to bring them on board and to get their feedback. The article took more than a year to write because we needed to agree. An agreement in these things is very, very difficult. But I think in the end, I'm glad that we managed. And what we wanted is that both the journals and the funding bodies take heed of this, because I think we cannot continue to fund and publish results and research that are claiming something that you cannot find in the paper. And I think we needed some standards in this field that is driven a lot sometimes by the hype of having human. I say sometimes that if you say that you've made a mouse embryo, nobody's going to listen to you other than your colleagues. But if you put there the adjective human, now you're talking, the echo chamber that you have is much bigger. So we have to be careful because that has impact. The funding bodies get very worried about this. They will start curtailing, as they have in some countries. Research in this area will start have limiting. So we need to show to the funding bodies, particularly, that we have really good standards and that the research is going to be done within rules that we ourselves have set up, that we are responsible. I feel, as a good friend of mine said, if we don't control ourselves, we are going to be controlled. So I felt that it was very, very important to show that we are serious about this work and that we know. And I'm very glad that I'm very grateful to my colleagues because it was a very good discussion. It was a very long discussion. As I said, it took almost over a year to do. But I hope exactly what I've said, that the funding bodies will see that this field is young, but is serious, and that we know what we are doing. No, we also discussed the issue of naming that it's a smaller one. But I think we have to recognize, as you yourselves have pointed out earlier, this can have big implications, and I'm sure it will for regenerative medicine. So we need to start at the beginning, setting up good standards for the field.
Yeah, and the other side of this coin is, making sure that the general public and the scientific community as a whole is just they're accurately understanding what these things are. The standards are, of course, important for those of us in the field who are working on these technologies, but the other part of it is accurately communicating what these things really are. I mean, that's part of the reason that we have a show like this podcast is, we want to be able to communicate the advances that are coming out in the stem cell biology field, not just to the scientific community, but potentially to the lay audience too, so they can accurately understand like exactly what these models are. There's such an explosion in these technologies that you and others are driving forward. And the last thing we want is for these technologies to be misconstrued as being something that they're not. You alluded to hype, and unfortunately, this field has had a lot of hype over the years. You know, we're doing our part. I know you're doing your part too. Social media can be a double-edged sword, as we all know. You're of course active on X, Twitter, whatever you want to call it, and discussing some of the recent advances in our field. Could you actually talk a little bit more about just the importance of science communication, the importance of accurately and appropriately conveying these new advances in embryo modeling to the public and also to the scientific community? How do we get that dialogue right so that people can actually correctly understand what we're doing without jumping to these wild, hyped up conclusions about this technology?
I have to say that as our listeners will understand, when I was working with fruit flies, I never thought that it was very important to communicate to the public that we were working with fruit flies. And I think that was obvious. And I never thought when I started doing developmental biology with the cells that we would find ourselves in this situation. But it is very clear that as you are approaching very early stages of development, and particularly the minute you start dealing with human models, you are touching very sensitive issues. And I object to people that say, for example, that the models that we have can help fertility or human reproduction, because I think while that possibility is in the horizon, I don't think what we have now it's appropriate for that. We have a lot to work. I think the blastoids are doing very well. To me, the blastoids, particularly the work of Nicolas Ridron, is very important. I think he's very sensible in piecing. Why is that? Because we would run into the same situation that the stem cell clinics have run over the years of promising, of over-promising and underachieving. And in the case, I don't have to tell you in the States these days where the word embryo, IVF, they can human reproduction, female fertility, it appears all the time. We have to be serious about what we do, because what we are talking will very easily permeate all that area and all those voices. And I think we have to show that we are serious about what we do, and we care more about what we do than about publishing a headline or publishing a paper. So it is true. I mean, together again, we published a year ago in Nature Cell Biology, who is really having a very responsible and very positive attitude towards the field, another commentary on how to communicate the development of embryology to the press. I think that this is very, very important, because as I say, in the end, this impinges in policy and this impinges in funding. And the two, particularly in this area, are very, very related. And if we show that they're irresponsible, and if people see that we are not serious, we are going to lose the possibility of developing this field to the potential that it has. I think that's the how to communicate it. Well, you have to learn and you have to be responsible and work with people. And that, I mean, in Europe, I'm not sure in the States, but in Europe, the UK, of course, has always taken a lead in this field of embryology and reproductive biology, and they are doing very well in dealing with the stem cell embryo models. France and the Netherlands have also are doing good, and we are trying to bring them all together in Europe into some unitary framework. I don't know how much of this is going on in the States, but we are trying to reach out to our colleagues also there. As I say, it's very important that we don't lose the support and the credibility with the funders.
Yes, and the funders and the public, right? And I'm glad you mentioned the IVF thing, because that, I mean, you could call it kind of the canary in the coal mine or whatever you want to call it. It's an indication of where we are and maybe the direction that we're going. I would never have predicted that. It seems like we're going backwards. And you, as you said, with the terminology, and now we're talking about, personhood for embryos. It's not far to go before you have people protesting outside our labs because of the personhood, or just fundamentally opposing. We can, roll our eyes or whatever, but it's if the people believe it and feel it, it's real, and we have to both respect it and we have to take care, not to allow our work to be targeted, because there's I will roll my eyes and say that these things are not an appropriate extension of the science that we're doing. But why risk it? We need to get the work done for all the good, right? And then they can see how much there is to lose. And as has been the case with IVF, you can see plenty of people across both political spectra that are in support of IVF because it's real to them, Republican, Democrat, whatever. It's real to them, and so there's protections for it. And we need to provide that same practical protection, I think, for our work by not overpromising and being careful about the work that we do.
I think that the history of IVF is a very good example. If you look at it, it's a fantastic piece of developmental biology and early embryology, and it's a very good model for how we should be conducting this research, with rigor, with ambition, and with self-criticism.
Yeah, and I think someone who had tremendous insight and prescience, and who I'm going to say here is Anne McLaren. I really enjoyed your lecture at the ISSCR. It was a really excellent tribute to herself in her eponymous lecture, which I think is not always the case. Rightfully, it's hard to have such a comprehensive synthesis that was so heartfelt while also being expansive and relevant to the science that has become, really polarizing in recent years. What do you think here? Just segue. What do you think of the legacy of Anne McLaren's work is today, and this is because I think you have a really good understanding, a deep understanding of her way of thinking. How do you think she would have weighed in on the current state of research?
That's very kind of you, but in this particular field at the moment, she would have rolled her eyes. I'm very sure she would have rolled her eyes and say some expletives. Okay, I think in an English kind of way. No, I mean, I got to know her a bit at a distance. She was in Cambridge for a while until she had this accident that she died. She was a very tough woman with very, very high standards. I mean, and I think, as I say, I think maybe in those days the technologies were so limiting that you had to think of the experiments very, very well. Today, there is so many possibilities and so many. I think she would have definitely not liked the current scene. And in fact, in my thought, as I say, I'm trying to bring together the important the leading figures in this field through these writings and through meetings to try to help each other, because I think competition is important. I'm no doubt, but we cannot be vying for headlines. We have to be vying for making this field the field that we want it to be, which promises in regenerative medicine, in reproductive biology. But we need to work seriously. I think the blastoids are going to be a huge help to IVF. I mean, just before the ISSCR, I was at ESHRE, which many people here will know is the society founded by Bob Edwards. And there, I mean, it was clear that these people are starting to look at blastoids as a test of the fertility and to increase the fertility, to understand the fertility. So we have to be very serious. There is a lot of defects. We know that pathologies that happen during gastrulation. That's why we have to get the standards and not think that we want a paper in a high-profile journal, but we want a method that other people can use. To me, this is the difference between a science and art. Okay, I mean. Bernstein could conduct like Bernstein and only like Bernstein. Nobody could conduct it. Or the Mona Lisa is the Mona Lisa. If you do a model that only your lab can do, I think we are in trouble. I mean, the best tribute, the best accolade that one in our field can do is that other people can do it. CRISPR spread through the labs like wildfire. PCR in its time, spread through the. I mean, this is to me the important thing, that other people can reproduce it and improve it. And this was part, as Arun said before, of this commentary, to emphasize one of the things that we have to relate the conditions. And McLaren, as I said, would have rolled her eyes. I mean, no doubt.
And of course, another technology that initially was met with skepticism and eventually took the field by storm was IPS, right? Everybody was trying to get there, but it worked, and now everybody in the world can make it work, right? So, I mean, you mentioned how you met Anne McLaren in Cambridge, and
briefly, don't. I didn't have so many interactions with her.
Sure, but hey, you've had interactions with folks around the world, and this is such a global field in embryo modeling, and there's folks from every single corner of the world working in this area. I mean, you've yourself have traveled the world in pursuit of science. So from Madrid, you went to Chicago for your PhD. Then you were in Cambridge for a long time, and before moving back to Spain, to the University Pompeu Fabra as a research professor. So you've had a global journey, and you're working in a global field. And we're always fascinated by folks who are willing to travel the world for science, because we're stuck here in the US, right? Maybe you could give some words of wisdom for trainees who are hoping to follow in your footsteps and maybe travel the world in pursuit of science. What was it like?
I wouldn't advise them. I think every time this question comes up, I think there is one thing. I'm very happy to share my thoughts, but there is something that is very, very important, and that I think many of my peers of my generation sometimes don't see it. The times are very different. The times today are very different. So, I did a few things like many people, but they wouldn't work in today's world. I think in those days, science was a very small field, particularly developmental biology. So I think you could travel the world, as you've put it, in pursuit of these interesting questions in a much easier manner than you can today. I mean, you could get a postdoc, you could get a job. I mean, these are things that are very hard today, and there are very talented people out there. But with regard to what I've learned is that different countries have different ways of doing science. There is no question about that to me, And I've lived a lot in the UK, and I have to say I'm sometimes a bit of a sentimental person, and I miss the UK because I like the way they do a science there. I think one of the things they've been leaders in developmental biology for many years. My late friend Louis Wolpert has been a towering figure in the field of developmental biology. And I think one of the things that I've learned from the British community is that thinking about your experiment is very, very important. Providing context is very, very important. And that's something that I learned from them, and that I hope I can. Chicago was a very formative time. I did my PhD there, but I always wanted to work on developmental biology. And I think I arrived to England where genes were just becoming part of the picture. I mean, come from a generation where we didn't know that development was driven by genes. So I've been very privileged to see that. And of course, Cambridge has been a fantastic quadrant of great developmental biology. People sometimes forget that Bob Edwards did IVF in the University of Cambridge. I mean, this is also sometimes has surprised me that the university does not celebrate this enough. John Gurdon, of course, although he did his early experiments in Oxford, he worked for many years in Cambridge. So I think that the ability for me, it's been a privilege to be able to interact with these people and to learn. Coming to Barcelona was a personal decision. Barcelona, as some of you might know, it's a big hub of genomics and cancer research. In fact, you should come one day if you want. I'll encourage you. There is, and I am surrounded. It's a very good environment. I think Spain as a whole is a country that punches scientifically way above its weight, but Barcelona has made a very good investment in the biological sciences, and it's paying, and I'm very happy here, and we are trying to develop the research that we started in the UK. But about the advice to people, I wish I could say just follow your interest and your excitement and do good work, and things will work out. I think that's only a component. of what you have to do today. Today, it's a little bit more complicated. And I guess the best advice is don't lose that interest and that excitement in the face of the challenges that the world is putting to you. And always remember that science is a quest, it's not a job.
It's a journey. I like how you started that by saying I wouldn't advise them, because I often think that someone asked me the other day, they're about to have a kid, and I'm not that old. I mean, I have a 10-year-old, so you'd think I'd have, and a 15-year-old, but you'd think I'd have like still be in my recent memory. And I said it's a very similar thing. I said, my advice to you is don't listen to anybody's advice, because the reality is that every individual experience is different, but also, as you said, in a very practical way, the kind of generation time, the relevance of your experience on a practical, like, granular level is not really that valuable. But, your thoughts and your experience have tremendous value. And that piece at the end, the general advice of like, yeah, like, dude, it should be, it's not a job. It should be, a passion, all that general stuff, that is always true. But the particulars may be a bit of a challenge. So I think you should advise, and you have advised, and we appreciate that advice.
One of the things Dale and I do, and I feel very committed to, is to provide a link with the older generation, to tell these people that you are part of a journey that is older than you are. I think this is something that sometimes this is missed, that we think that things are instant. But the story of stem cells is a very beautiful one. And it comes from developmental biology. There is no question that stem cell is not something that appears all of a sudden in the late 90s. It's something that has a very long history. So that's, I mean, as you say, it's not general advice, but you have to listen to people. I think mentorship is very important today. It's more important than it's ever been, because the challenges that people face are huge, particularly the young people that want to do a science. And I think that's a very, very important thing, particularly when you find someone that is really talented and that has true ambitions, and those people are very precious, and you have to help them navigate in the world that you are today. I agree with you.
Well, I've learned a lot just from this conversation, I have to say. And one thing that brings out, it's true, the ambition, get the big paper, but as what's left for you, as someone who's been in the field for some years, is having something that's reproducible. What a great synthesis of the legacy of science. It's not how many Nature papers you have, but how many people can do what you found. So thanks again for sharing. Before we let you go, we had a couple kind of science peripheral questions. The first one is, I don't know, you've done enough for sure, but if you were not a scientist, what would you be doing with your life?
My big frustration is music. I would have liked to be a musician, but musical education in Spain is not incorporated in the schools. But if I have a frustration, that's the frustration. That's what I would have liked to do. Of course, you can be a scientist and a musician. No, as I told you, my wife plays the flute. She's very good, and she only makes me envious, because I think music is something that I love.
I must say, your fluency with language and storytelling is music to my ears. Sorry, I couldn't help myself. Finally, what is the biggest misconception about science that you would like to resolve?
You fast. I wonder how you're going to put it at the end. I think the biggest misconception, of science is that there is a truth. I think there are things that work, and I think this is something that I've realized over the last years. And I have to say that working with stem cells has made me realize of the difference between a truth and what works. I think that's also a contrast between biology and physics. It is very difficult to know, for me, what is the truth in biology. Very easy to work in physics. In fact, in a way, I've always felt that working with stem cells might lead us to that from that sentence from Feynman: that which I cannot build, I do not understand. I think there is a very good we can do wonderful things with CRISPR because we understand molecular biology in extreme detail. I'd like to get to that great detail with the cells and with the stem cells. If we build something efficiently, it's obviously because we are using something that we understand to build. Truth is something else. And sometimes these days I've forgotten about what is the truth.
Yeah, well, what I've learned from my 15 years of marriage is that the truth is irrelevant.
My friend, you're getting into a different territory there. I have almost 40 years of marriage.
Oh boy. Well,
I have more papers than you on that, okay?
Well, if you're still happy, then maybe that's better than truth.
The papers are unpublished. Anyway, great talking to you guys. Thank you very much. And I keep on listening to you and learning. Okay?
It really has been our pleasure. Thank you again so much for sharing with us and our audience. All right, you guys, that brings us to the end of this episode. What a delight it was. Truly one of my favorite people in science. Not that I know him, although I do now, but just a great storyteller. And I think an icon worth looking up to for all the trainees and all the adult scientists among us. Alfonso, thanks so much again for joining us on the show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on X at StemCellPodcast or via email at info@stemcellpodcast.com. There, you can leave feedback or suggest guests. Until the next one, I think we have an off-schedule episode coming in a week. So look forward to that. Until then, thank you guys so much for listening.